hello. Thank you for attending. I'm really happy to be starting the symposium today. Is everything working well? Sorry, that's all right. Okay. Thank you. Okay. Happy to be starting the symposium today, and I'm looking forward to hearing from all of you today. I'm going to go ahead and start by thanking Susie and BCBC and BTI for this internship. It's been a wonderful experience and something I definitely won't forget. So what have I been doing with my life for the last 10 weeks? This is progress towards understanding the interplay between TYLCV resistance and heat tolerance. And as we mentioned just a moment ago, TYLCV is tomato yellow leaf curl virus. So why is this important? Climate change, a global crisis. As temperatures increase globally, arid regions are seeing record-breaking temperatures, which trigger heat stress and reduce crop yields. Many agricultural crops are moving toward increasing heat resistance in an effort to maintain agricultural viability as temperatures increase. And stressed plants not only produce lower yields, they're also more susceptible to pathogens such as TYLCV. This is a graph from the global surface temperatures by NASA. This is most recent, and you can see how starkly and quickly that is going up. So this is something that's really important and going to be super key to continuing to have really good crop yields into the future. So tomato yellow leaf curl virus. This is one of the most common viruses to Selenium lycopersicum, or just common tomato plant. It's one of the most prolific viruses. It's aptly named in that it does exactly as it says it does. And you can see that here in this image. So these are 2 healthy plants and 2 infected plants. And these are on the extreme end. This is an image from the grant proposal for this project. But this is a very severe case, but gives you a good idea of some of the symptoms and how this actually affects the plant. And this is a very heat tolerant line. And this is one that's also very susceptible to the virus. So that's part of the issue that we're focusing on today, is that some of the most heat resistant lines are also the most susceptible. So how do we figure out how to keep them alive? So this is a basic phenotyping table. I'll probably come back to this throughout my talk. But it's just a couple of names of lines to clear up what the phenotypes actually are and when I'm talking about them. So 2553, that was our reference genome. And that one is a heat tolerant and virus susceptible. So that's pretty much what we saw on this slide here, one of these. So 906-7, that's virus resistant and inducibly heat tolerant. So basically it's when it's infected with the virus, it develops heat tolerance. And that's what we're focusing on, looking at that interaction. And then 906-4, that one is just virus susceptible and heat susceptible. So it's kind of, yeah, not the most agriculturally important one, but we'll move on. Okay, so the virus susceptible lines, how does the system work? Essentially, what we have is 2 stressors that are combining to create the major issues we're seeing. We're seeing heat stressed plants that are then being infected by TYLCV, and we're seeing the stunted growth that we observed earlier. So the 906-7 line, the heat and virus resistant, what's happening is virus infection, then they develop heat tolerance and can tolerate more heat stress, and they're actually growing healthfully. So the research goals, we're looking for locus or loci linked to this heat tolerance and virus resistance that we're observing. We're doing that by looking at SNPs, so small nucleotide polymorphisms, or single nucleotide polymorphisms. Using an array of tools, we're going to make some figures in R, and our results today are based on allele frequencies from this data. So a little bit of background on the Genome Assembly. So this is important because this is the 2553, our reference genome. This was done by, put together by Jing. So thank you, Jing, so much. That was the basis for all of this. So couldn't have done it without you. So 2553 is the heat tolerant virus susceptible line sequenced using nanopore sequencing. And part of the main thing we're focusing on this page is this config N50. That's a good one, especially for nanopore. Yeah, it's just showing quality of the genome. Genome Assembly, so this is continuing with that. This is just a graph of Genome Comparison, which Genome is just a program that shows completeness of a genome. So we have Arabidopsis, and a couple others. And then this one here, this green guy, is our 2553. And as you can see, comparatively, very little fragmentation and very little missing, which is good because we're going to use it to compare. So if it's not there, we can't compare anything. Okay. So how did we get our genomic SNPs? This is what I spent a lot of time on. Basically, this is a basis of our SNP calling pipeline that we built. So using these programs, and this is just a little bit about what they did. So we looked at regions of low SNP density using this throughput pipeline, high throughput pipeline. And we were looking for variation in heat resistant, virus resistant lines. There'd be high SNP density in regions which may indicate that they're producing, or what's causing this observed phenotype. And the same region should have high SNP density compared to heat and virus susceptible lines, because that would further indicate the regions are involved in the system in question. So after we got through all that, our results. These are still a little bit messy, but we're starting to see some stuff. So what we have here, this is the reference, and this is this is the R line that I mentioned earlier, the 906-7. So as we can see, mostly in this region here, very few. This is on chromosome 11. There's not much going on down here, but you can see a huge difference in the number of SNPs in this line. So this is just the reference and the actual sample. So this is not conclusive, but it is kind of guiding us toward where these things or what might be going on. Again, this is on chromosome 11, and it may be of further interest because this this region was mentioned in described in Frontiers in Plant Science, Yan et al. 2018, that this may be a TYLCV resistance region. So that's looking good. There's definitely still some more like data processing to do, and we have a couple of more samples that we didn't get data for quite yet. But yeah. Okay. So genomic data only tells us half a story. So the genome is only consequential where it begins to be transcribed. And this is the focus of the second half of this project. So RNA sequencing will allow us to compare not only what is there, but what's actually being transcribed under plant stress. So to accomplish this, we started by extracting RNA using the QIAGEN RNeasy kit, which I think is a really cute name and not as easy as it sounds. So then we went ahead and sent that for quality control at Cornell Biotech Resource Center. Really appreciate them doing that. And what we got is, I thought I put it in here. There it is. We'll get there. And then we're going to go ahead and this part is in the future. So we're in a time machine and we're going to send it for 3' RNA-seq. Okay, so what did we actually extract? So these were samples sent in RNA later from our collaborators. I didn't name these, so they came like that, but here's what they are. They called this one TOY. It's tolerant, heat tolerant, and virus tolerant. And then Validity, which is that's the very heat tolerant, extremely susceptible, virus susceptible. So that's the like 2553 line that we've been looking at. And then we have a couple of other heat tolerant and virus resistant. And these ones were exposed to subjected to heat stress at 0 minutes, 30 minutes, and 3 hours. Each had 3 replicates, and then each had 3 replicates inoculated with TYLCV and 3 replicates without. And samples were taken at 10, 15, and 20 days after inoculation. And this adds up to a total of 72 samples. And I found out that 72 is divisible by 3. So figure that one out. So the results of our QC. So these are just some of the stuff that, just like a few test samples that I threw in to see if what we were producing was good RNA. And so what we have at the top here is a whole mess. This is just DNA. And that's actually something that we're working on to get, we need to treat it with DNAs before we send it off for 3' RNA-seq to get rid of some of that. But this is really good RNA. This is what we're looking at here, these lines. And then of course, empty, but that happens. There's just nothing in there. I don't know where it went. I dumped it down somewhere. Okay, but yeah, these two strong bands are the RNA we're looking for. But unfortunately, due to time constraints, I'm not able to finish prepping the RNA for RNA-seq before the end of the program. So that moves on to here, work for the future. So what are we going to do to continue this project? Next steps are 3' RNA-seq. So we need to clean up those RNA samples and send them off to for RNA sequencing. And then we're going to go ahead and run differential expression analysis on that data that we get back. And then this genetic marks and genetic mapping. This actually is these are going to be built based on the single nucleotide polymorphisms that I have been working on during this project. So those will be built using those and then used for genetic mapping for a mapping population that is currently in the works. So we don't have that yet, but we will. Yeah, so that's what's happening in the future with this project. And finally, thank you to my supporters and contributors, BTI for this internship, Susie for being wonderful, just a wonderful guide and mentor. Jacob Landis, he's in Chelsea Specs lab. They, for some reason, let an undergrad run wild in their lab. So I did that, and Jing, of course, put together the reference genome, so it wouldn't have been possible. Yeah, thank you so much. I can go back to slides too if we need to. Yeah. Essentially, yes. Yeah, because just because the gene is there doesn't mean it's really doing anything. So yeah, that's just looking at what's actually happening in the plant when it's under stress. Yeah. Mm-hmm. Right. No, so those ones are just, yeah, they're just virus tolerant. So when they're treated under stress with no virus, they don't perform particularly well. So when they're inoculated with the virus and then subjected to heat stress, they do much better. And that's, yeah, we're looking at the actual gene, the genes that are interacting to create that phenotype. Then that's part of what makes this project a little bit different than what's been done, just because it's like we've observed this in the greenhouse. This has been like something that's been common for plant breeders, but we don't actually know what's happening or why. So that's what we're trying to figure out. Yeah.
Are there specific harvest genes that induce the heat tolerance?
That's kind of what we're looking at right now. There is like a little bit of suspicion that there isn't like an actual gene explanation. It's just sort of like there's an assumption that the virus like reduces the heat stress on the plant basically so that it can better proliferate, but it doesn't say anything about the genes. So that's what we're trying to figure out. Yeah.
Does the virus itself still have negative effects on the plants with virus heat tolerant plants? Just purely beneficial?
Yeah, I don't actually know. But that said, it's virus resistant. It is not like virus proof. Yeah. So I mean, at some point, I'm sure it'll have negative effects. But yeah, it's mostly fine. Yeah. Mm-hmm. Yeah. Yeah, it's not just going to be chromosome 11. That was just one of the only data points we had so far. There's still a few things we're looking at. We still have to call snips on some of the heat resistant lines and compare those. So I'm sure that there'll be more genes of interest that we'll be looking at.